The diaphragm is not the only respiratory muscle. So one thing I need to do to preface you for this discussion of respiratory muscles and how active and passive respiration works is to distinguish between quiet breathing and forced breathing. So we will talk about regulation of the respiratory system and these muscles by the nervous system a little bit later, but everybody's had this experience, so I want to describe it. Although I can change my respiration voluntarily, like I can modulate my breath when I want to speak, or I can decide to scream, or I can fake cough, those are all examples of forced breathing. Normal breathing, quiet breathing, is effortless if you're healthy and does not require that you think very much about it. So we have both conscious and unconscious aspects to our breathing. And that's why, for example, if I'm thinking about something else, I don't suffocate to death while I'm thinking about it. So I don't have to constantly be thinking, inhale, exhale, inhale, exhale. Normal breathing at rest is mostly due to the diaphragm. So this is why I was stressing the importance of the diaphragm and the phrenic nerve earlier. The remaining is due to the action of the external intercostals, but they don't contribute very much under normal conditions. Forced breathing is more complicated. So you have to use the muscles of inhalation that you normally use, but to a greater extent. So send more action potentials down that motor neuron, and also engage accessory muscles. Anything that pulls your rib cage up or laterally is going to be an accessory muscle of inhalation. So your sternocleidomastoid pulls your clavicle and the manubrium of your sternum up. The scalenes also pull on the clavicle. Your pectoralis minor pulls your ribs up and out. And the serratus anterior pulls your ribs outward laterally, opening them like a book. So all of those are going to be muscles that you're employing when you are very intentionally and consciously taking a big, deep, powerful breath in. The forced muscles of exhalation are the internal intercostals, which compress your thorax, and the abdominal wall, which puts you into sort of a crunched position, which further decreases your thoracic volume. So here's another example. Again, quiet breathing is, inhalation is active. So if you were to, say, put a electrode on the phrenic nerve and observe what was happening in the phrenic nerve during inhalation versus exhalation, you would see a spike train of action potentials happening during inhalation and then a quiet moment during exhalation. So you don't actually have to do anything extra to exhale. You just have to not inhale. So diaphragmatic or deep breathing is typical at rest. Costal or shallow breathing happens when there's increased abdominal pressure in the form of fluids or masses. So this is something that you would see mostly in a pathological context. Forced breathing requires extra muscles, as I discussed previously. So these are ones that you're going to have to actively choose to engage to voluntarily or consciously change your breathing. So whereas quiet breathing originates signal-wise in the pons and is regulated by the medulla, forced breathing initiates in your precentral gyrus. Remember that? That's your primary motor cortex. So the signal to perform voluntary respiration originates in a different place than the signal to perform passive respiration. So before we move on, I want to describe a pathology that relates to this because it's interesting. And it has an interesting name as well. So this pathology is called, and let me grab my pen instead of my laser. Ondine's curse. Isn't that a fantastically dramatic name for a pathology? I think so. So the Cliff's Notes version of Ondine's curse is it's a legend wherein somebody was cursed to not be able to breathe while they were sleeping. So we have the luxury of being able to passively respire without any conscious effort while we are asleep. In Ondine's curse, that pathology creates a situation where the signal to breathe passively and quietly does not originate from the brainstem. And so exhalation and inhalation are therefore only conscious. So they require active effort on the part of the individual. So this is a relatively unusual pathology nowadays, but it used to be more common, and it was more common in the time when polio vaccination was not yet developed. So poliovirus affects motor neurons, which is why it results in inability to use certain muscles, and it can also affect the brainstem such that the signals to inhale via the phrenic nerve are not automatic and not functioning. So if those individuals want to breathe, they have to activate their premotor cortex and motor cortex to do so, meaning that breathing is only ever conscious for them. So if you're curious about that, look it up, but it's pretty interesting. These are the respiratory rates and volumes. And this is a reference chart, right? So this is just an example. Obviously, if you used a spirometer on a patient to estimate these things, your readout or trace would look much different. So to give you an idea of what's happening here, on the y-axis is volume in milliliters, and on the x-axis is just time. So because this is an example trace, time is unitless here. What matters more are the volumes up and down on this chart. So tidal volume is what we call the volume of air moving in and out of the lungs at rest. So when you're not actively trying to moderate your breathing, you are just sitting there breathing quietly and passively. So for most folks, tidal volume is about 500 milliliters per breath cycle. So half a liter. Now, take a quiet breath out. Oops. And then after you're finished quietly breathing out, keep going. Go until you can't expel any more air from your thorax. Whatever volume of air you manage to expel is the expiratory reserve volume. So the reason this doesn't have a number here, as you can see, is because it's going to vary from person to person, right? It depends on the level of development of your respiratory muscles, and it depends on your lung compliance and your size, etc. So if you look over here at this chart, we have ERV. For males, it's about 1,000. For females, it's about 700. So these are reference values that have been taken from averages of many individuals. So that's why there's no number here, because it depends. Inspiratory reserve volume is what happens when you passively and quietly breathe in, and then you keep breathing in and in and in and in and in and in. So keep going. So again, inspiratory reserve volume for males tends to be larger than for females, and that is because, on average, males have a larger thorax and therefore more expansive ability. If you look down here, so before I move on to this stuff, let's talk about these. If you note, down here is 0, right? None of these curves ever make it to 0. What that means is you are not wringing out your lungs like a sponge every time you breathe out. That would cause alveolar collapse and not be adaptive at all. So residual volume is the amount of air left in your lungs after you exhale, and that keeps your alveoli open. So the minimal volume is anywhere between your expiratory reserve volume and 0, or excuse me, residual volume is between your expiratory reserve volume and 0. And then the minimal volume, this is the absolute minimum required to keep your lungs open. This is not physiologically possible to reach voluntarily. So this would be an example of like operating in a low pressure environment or something like that. You don't really get below this before your lungs collapse. Your total lung capacity is from 0 milliliters, so not inflated, all the way up to the top of your inspiratory reserve volume. Your inspiratory capacity is how much you can voluntarily inhale if you want to. So the difference between inspiratory reserve and inspiratory capacity is reserve is in addition to tidal, and inspiratory capacity is IRV plus tidal. Vital capacity is the total volume of air that you can voluntarily and strenuously take into and breathe out of your lungs. And vital capacity is perhaps the most telling metric for clinicians, because reduced vital capacity, you can subtract milliliters from either side. So either you can inhibit inspiratory reserve in some way, or you can inhibit expiratory reserve. So either of those things will change vital capacity, but which end of the arrow is affected means different things clinically. So for example, if I saw a reduction in vital capacity and I knew somebody had a history of lung issues, maybe they had a history of tuberculosis, I would assume that collagenous scar tissue from TB infection was reducing their lung compliance and therefore chipping away at this top part of the arrow. If I saw a patient that had a relatively normal vital capacity-ish, but they had a reduced expiratory reserve volume, that would imply to me that they had pulmonary obstructive disease, meaning that their airway was narrowed and therefore it takes them a lot longer to exhale, which reduces the expiratory reserves. Those are just some examples. And then the functional residual capacity is after you passively breathe out, the difference between that and zero. So I would get familiar with this chart and be able to compare and contrast these arrows. So for example, if I ask you on an exam question, which two things would you add together to get inspiratory capacity, you would need to come up with IRV and tidal volume. Hopefully that makes sense. Now we are getting into gas exchange. So we have some more laws. So Boyle's law was pressure is proportional to the inverse of volume. We also have Dalton's law and Henry's law. So Dalton's law and Henry's law are the physical laws governing how and whether dissolved oxygen and carbon dioxide can move between the air and our blood. So these laws govern effective gas exchange via diffusion and therefore respiratory function. So let's take a look. Dalton's law is about partial pressures. So atmospheric pressure, which you can measure in atmospheres, so 1 atmosphere, we're going to stick to millimeters of mercury because that's what we've been using in class. So atmospheric pressure is 760 millimeters of mercury. But our atmosphere is not only oxygen. Our atmosphere is a gas mixture. It's a mixture of nitrogen, oxygen, water vapor, and carbon dioxide, as well as trace other gases, but those are the big contributors. In fact, nitrogen is way more abundant in our atmosphere than oxygen is, for example. So all Dalton's law does is state that if you have an air that's mixed, like our atmosphere, the total pressure of that air, the 760 milligrams that you're seeing here, is the sum of each of the contributions of the individual subset gases. So pressure of nitrogen plus pressure of oxygen plus pressure of water plus pressure of CO2. So you have to add those up to get the total. Fortunately, this is pretty easy to calculate if you understand percentages. Not that hard. So 760 milligrams of mercury, or millimeters of mercury, is 100% of an atmosphere. So if you know that, for example, oxygen is 20.9, so about 21% of the total, all you have to do to figure out how many millimeters of mercury oxygen is contributing is multiply 21% times 760 millimeters of mercury. So if you wanted to perform that calculation on a calculator, you would write 0.21 times 760, and it would spit out 159 millimeters of mercury for you. So that's how you figure that out. So the partial pressure of oxygen is one of the important driving forces for exchange between alveolar air and our blood. Alveolar air is moist and contains water vapor, and that's because of the conducting division of our respiratory system. So think about the inside of your face and pharynx, so your nasal cavity, your oral cavity, your larynx, which is your voice box, and your trachea. Those all have wet inner surfaces, right? When you breathe in, the airflow inside of your nasal cavity, nasopharynx, larynx, etc., the airflow in those structures is mostly not laminar, it's turbulent. And that means that the gas molecules that you are breathing in are hitting the wet walls of your inner spaces and picking up water from them. And that helps keep alveolar air moist. That's important because, again, you can't exchange gas across a dry interface. Your alveoli have to be moist on the inside in order to exchange air. So using the same equation, but correcting for water vapor that's picked up on the way down, results in a partial pressure of oxygen in alveolar air of 100 millimeters of mercury. So if we go back and look at the PO2 of atmospheric air, we have 159. So water vapor alone takes away 59 millimeters of mercury from the partial pressure of oxygen in alveolar air. If you've ever been in a very, very humid climate, either for vacation or you've lived there before, say the southern United States or parts of Asia, etc., or places on the East Coast even, you know very well how much harder it can be to breathe in humid air versus very dry air, right? Part of that is because water vapor depresses the partial pressure of available oxygen and takes its place. So partial pressures in general, we've got sort of a summary table here. So nitrogen, oxygen, carbon dioxide, and water vapor are all represented. I'm not going to read this chart to you because you're capable of reading it by yourself. Understand that where we have blanks, it just means millimeters of mercury. We're just not repeating it. Alveolar air is an important one, especially when you compare it to inhaled air and exhaled air. So the reason that these are bold is because you're supposed to notice the difference in alveolar air. So 100 millimeters of mercury in alveolar air for O2, and CO2 is only about 40. And by the time you exhale, 116 millimeters of mercury and about 28 of CO2. So if you're wondering about these differences, that's what we're going to start to build up as we continue looking at the forces driving respiration. There's a question on the chat, and that is 100 millimeters of mercury replaces 159 as a constant. Not in all cases. That is specific to alveolar air. So if I were to ask you a question about that, I would specify in the wording of the question whether I was talking about 159 or 100, and I would say alveolar air, or I would say inhaled air. So you would be able to tell based on the wording of the question which you were supposed to use. I'm not going to leave that up to being nebulous or undefined. Does that answer your question? Awesome, good. Also, thanks for asking. I was beginning to suspect that maybe no one was hearing me because there was nobody talking on the chat. Okay. So, Henry's law is the next law, and I mentioned that you need a fluid interface in order to perform gas exchange. And that's true, it's required. So you have to have oxygen molecules dissolving into water that's coating the inside of the alveoli, and then dissolving from that water into the blood. So this is where Henry's law comes in. So Henry's law is about driving gas molecules into solution and how pressures and volumes relate to that. So the amount of gas that will dissolve in a fluid is driven by two things. One is its partial pressure in a gas mixture. The other is its solubility coefficient. So it turns out that due to the chemistry of water, the way it hydrogen bonds, its polarity, its properties as a solvent. Different gases have different solubilities in water. So the best example I can think of for this is soda. So at my house, I drink a lot of seltzer water like LaCroix and such. I think we all know what happens if you open a can of carbonated water and then you go away for a while. When you come back, your soda's flat and then you're super bummed, right? You're like, oh, now it's just kind of water that tastes vaguely fruit. So why is that? What happens? At the cannery, the gas mixture containing carbon dioxide is put into the can along with the fluid under high pressure. Fortunately, carbon dioxide is fairly soluble in water, and it doesn't take a whole lot of pressure to force more carbon dioxide into solution. If you keep the pressure stable by controlling the volume, so you put a lid on the can and you seal that lid and you don't let anything in or out, as long as the soda can is unopened, a significant amount of that carbon dioxide is going to be in solution at equilibrium for that pressure. So equilibrium states for air liquid interfaces and gas dissolved in them depend on a couple of things. One of those things is temperature, the other thing is pressure. So at a particular temperature and pressure, there is an equilibrium between number of gas molecules dissolved versus number of gas molecules in air. That equilibrium state changes once you open the can. So if you increase the pressure of a gas mixture in air and you increase it against a fluid interface, you drive more of those particles into solution. Note that that increase of pressure here is caused by a decrease in volume. So once that happens, you have no net movement between air and water after a while, and you reach equilibrium. So what happens if you take this equilibrated can and then you're like, I wish to consume this beverage, I shall open it. Now, the gas has to reach a new equilibrium because there is no longer the pressure driving molecules into solution that there once was. So more gas molecules are free to dissolve out and aerosolize and join the gas form again. So once the can of soda has its pressure relieved, the bubbles form, which is why it tastes effervescent, and the dissolved gas leaves the solution. But what happens after a while, of course, is that molecules are leaving at a higher rate than they're entering, which is why your soda goes flat. Henry's law in the body is important because it relates to pressures, specifically partial pressures, and specifically also to solubility. So oxygen and carbon dioxide have different solubilities, and therefore they behave differently under the same atmospheric pressures and partial pressures. So Henry's law in the body allows the oxygen in the alveolar air, so remember that's 100 millimeters of mercury, to diffuse into the surfactant and then cross this space. And the converse for carbon dioxide. So partial pressure of carbon dioxide is higher inside of the alveolar air than it is in the blood that's passing over the alveolus, or excuse me, lower, and then that means the carbon dioxide moves from the blood into the alveolar airspace. So here we have a diagram. It's a very simple diagram separating the systemic circuit from the pulmonary circuit. And this is the focus of our attention for internal respiration. So the next couple slides are going to look at this in a little bit more detail. So if you pay attention to alveolar air, so here we have external respiration. We've got our 0.5 micron respiratory membrane. Partial pressure of oxygen in the alveolar air is 100 millimeters of mercury. Partial pressure of oxygen in the venous blood coming in from the pulmonary artery is only 40 millimeters of mercury. So, obeying the laws of diffusion, oxygen is going to diffuse from an area of high partial pressure to an area of low partial pressure, and that's how we load oxygen into our red blood cells. The opposite is true for our carbon dioxide. So Partial pressure of carbon dioxide in oxygen-poor blood is 45 millimeters of mercury. Partial pressure of CO2 is only 40. So even though there's a smaller difference in pCO2, it still diffuses out into our alveolar air enough to sustain survival. Now, you might be asking the question, why does the difference magnitude between the two differ so much? So why is oxygen's partial pressure difference so much more than carbon dioxide? And why does carbon dioxide still get efficiently scrubbed out of our blood and into the air? The answer has to do with a couple things. One of those is chemistry. The other is solubility difference. So carbon dioxide's solubility in fluid compared with oxygen's is different, so the driving forces are slightly different. So here's what this looks like in the systemic circuit. So when your capillary beds are exchanging oxygen with metabolically active cells and tissues. So we've got partial pressure of oxygen is about 95 at the arterial. Partial pressure of CO2 is 40. In a metabolically active tissue, oxygen is being constantly consumed by mitochondria, so the partial pressure of oxygen is low compared with the blood. So oxygen is going to diffuse down its partial pressure gradient from an area of high partial pressure in the blood to an area of low partial pressure in the tissues. The opposite is true for carbon dioxide. So metabolically active cells are going to be actively producing CO2, which means that the partial pressure in a metabolically active tissue is going to be higher, and it's going to diffuse down its concentration gradient from an area of high partial pressure to an area of low partial pressure, which is about 40. So again, smaller diffusion difference, but the solubility difference between oxygen and carbon dioxide accounts for efficient exchange. So let's talk about gas transport. How does it work? So we're zooming in now, notice that? So it's not even as simple as looking at the tiny respiratory membrane. We also have to think about red blood cells and what's in them. So I mentioned a difference in solubility, and that really does matter here. So only about 1.5% or so of the oxygen that you take in from the atmosphere is dissolved in your plasma. So that's why you can donate plasma and not die, because if you donate plasma, you're not donating red blood cells. And that's also why there's a limit to the amount of red blood cells you can lose before things start to get very hairy medically for you. So 98.5% of that is bound reversibly to hemoglobin. So what do I mean by reversibly? That means that oxygen can hop onto hemoglobin under certain chemical conditions and hop off under others. And that's really important. What do you think would happen if hemoglobin bound to oxygen irreversibly? What consequence would that have on respiration? I'll give you guys a minute to think about it and answer. Okay, enough time has elapsed. I'm going to answer for you. So if the word reversibly here was changed to irreversibly, what that would mean was, once your hemoglobin had picked up an oxygen, it would never ever let it go. Which means that you would have a permanent state of oxygen-rich blood, but it wouldn't do you a damn bit of good because you could not drop that oxygen off at your tissues, so you'd die. So the reversible binding is important, and I'm driving that point home very intensely because anything that challenges binding affinity between hemoglobin and oxygen changes the efficiency of your respiration, and sometimes that becomes clinically relevant in a very big way, in a way that can potentially affect patient survival and health. So for those reasons, you should be concerned about this. So factors that dictate the reaction above. So hemoglobin plus oxygen binds to become oxyhemoglobin and also can dissociate into separately hemoglobin and oxygen. Plasma pH is a big deal. So how acidic or not acidic is your plasma? Temperature is another one. And metabolic activity inside of the red blood cell. So bisphosphoglycerate, which we abbreviate BPG, this is a byproduct of glycolysis. It actually affects the degree to which hemoglobin and oxygen are bound. So this brings us to some curves and graphs that tend to scare students. Mostly, I think, because of this phenomenon I call chart anxiety, where graphical representations of data just like immediately cause some people to freak out, even if they haven't looked closely at the axes. And I totally get that. It's mathy, it looks scary on the surface, but I promise you they're easy to understand. So the oxyhemoglobin saturation curve, and notice this is talking about at a pH of 7.4, so at physiologically normal blood pH. This curve exhibits a property called cooperative binding. So who can tell me how many oxygen molecules a hemoglobin can bind? Anybody? 4 is correct. Thank you. So 4 oxygen molecules. Note that I'm using the word molecule very on purpose. So if I asked you how many oxygen atoms a hemoglobin could bind, you would need to say 8, because oxygen molecules have 2 oxygens per molecule. So there's a big difference between molecular oxygen and atomic oxygen. So one thing to understand is that hemoglobin is a tetramer. So there's 4 globin chains, 2 alpha, 2 beta, and in each of those globin chains, there is a heme group with a ferrous iron at its center. It is absolutely not the case that all 4 oxygens jump onto the hemoglobin at the same time. So a hemoglobin will pick up one, and then pick up another, and then pick up another, etc. So what cooperative binding is the idea that once a hemoglobin molecule has bound its first oxygen, its conformation changes a little bit, and it increases the likelihood of the second oxygen to bind. And that increases the likelihood of the third oxygen to bind. So what that means is the binding of the first oxygen molecule to a hemoglobin promotes the binding of the second due to the protein kinetics, so the changes in protein shape that happen when oxygen binds to hemoglobin. So on the y-axis, we have percent saturation of hemoglobin. So this is just of all of your hemoglobin, what percentage of that is carrying O2. And on the x-axis here, we have the partial pressure of oxygen in the blood. So that's it. We're just plotting how much binding is happening versus PO2. So you can see that there's a positive relationship such that the higher the partial pressure of oxygen, the higher the saturation of hemoglobin. And the reason the curve is not linear is because of cooperative binding. So that's also important. So if there was no cooperative binding, if there was no relationship between binding one hemoglobin and the likelihood of binding the next, if that was true, this curve would just look like this. It wouldn't have this exponential growth component in the beginning. So when you compare the difference between this line and this curve, the significance of that becomes very obvious, right? So the cooperative binding really does matter. It facilitates oxygen saturation of hemoglobin, which is important. So when you go to the hospital, I hope no one has to, obviously, but if you do and you're in the ER or admitted, they put a little doohickey on your finger called a pulse oximeter. What that machine does is shines a red light through your finger, and a sensor opposite your finger, so it's on the other side of your finger from the red light, the sensor detects how much of that red light gets through. So pulse oximeter estimates your oxyhemoglobin saturation by estimating redness, because the only time when that redness happens is when oxygen is bound to hemoglobin. So that's how a pulse oximeter works, the quick and dirty version. Here we have the oxyhemoglobin saturation curve related to pH. So notice in this last slide, we said at pH 7.4. So that's that middle curve here. pH changes the binding affinity of hemoglobin for oxygen. And this should say affected with an A, not effected. It's a pet peeve of mine. I need to fix this. Mark spelled it wrong. He used the wrong one. So just real quick, if you want to make sure that you're writing competently, affected is how you use that word in this particular context. So pH changes hemoglobin affinity for oxygen. Here's how. As pH decreases, O2 release increases. What that means is the more acidic your blood is, the lower the binding affinity of hemoglobin for oxygen. So an increase in acidity, decrease in pH, encourages your hemoglobin to let go of its oxygen. Now, that might seem bad, right? Like, oh no, acid, my hemoglobin is not going to be able to hold on to my oxygen very well. It's actually very important physiologically, and this is one of the reasons why pH is managed by your body so closely. If a tissue is metabolically active, it's going to be producing a lot of carbon dioxide, right? Because carbon dioxide is a byproduct of cell respiration. Your body is also full of water. So if you have, imagine a cell, and your cell is surrounded by red blood cells with hemoglobin, and this cell is really metabolically active, so it needs a lot of oxygen. So if this cell is demanding a lot of oxygen and spending that oxygen as a final electron acceptor to do cellular respiration, so Krebs cycle, oxidative phosphorylation, it's going to be pumping out a lot of CO2. CO2 is a good proxy for amount of metabolism happening. That CO2 is going to diffuse away from the cell and enter water, either inside of the red blood cell or outside of it. That CO2 is going to combine with that water to form carbonic acid. That carbonic acid, it's an acid, right? So acids are defined as substances that donate protons, this guy, in solution. So because it's an acid, that proton is going to hop off, and then we get H+, which is the units of acidity. So the more of these there are, the lower the pH, and bicarbonate. So the carbonic anhydrase reaction that I'm showing you down here is one that you were first introduced to in Biology 160, and it's still important now. So this is called the Bohr effect. The Bohr effect is just stating that when you change pH, the curve shifts. So if you look at what percent of hemoglobin is saturated at a given PO2, so let's say at 20 millimeters of mercury of oxygen, 60% of hemoglobin are saturated with oxygen. At 20 millimeters of mercury for a lower pH, we got about 25. That's a pretty big difference. Because of this phenomenon, metabolically active cells naturally facilitate unloading of oxygen more in their surroundings because they're producing more acid. So it automatically holds that metabolically active tissues can suck up more oxygen because the area around them is more acidic, and therefore the binding affinity of oxyhemoglobin or hemoglobin for oxygen is lower. Same is true for temperature. So at any partial pressure of oxygen, an increase in temperature decreases the affinity for oxygen. If you think about environmental events like fish kills, if you guys are familiar with this, you may have seen them in the news. Sometimes fish kills are results of plankton and other stuff consuming all the oxygen in the water. A lot of times fish kills come from heat waves or masses of warm water. So at any PO2, an increased temperature results in decreased oxygen affinity, and at any PO2 in water in general, water hangs on to oxygen less well at higher temperatures than lower. So for hemoglobin, the increase in temperature causes the hemoglobin molecule to flex and expand faster, because all proteins breathe. They subtly shift conformation. The higher the temperature, the more they do that. And that increases the likelihood that the oxygen is going to hop off. So same deal here. If you look at a very high temperature, so 38 degrees C is just north of body temperature, 43 is considerably higher. So the saturation of hemoglobin at 40 millimeters of mercury is a lot lower. It's about 50-ish than at 38 degrees Celsius, in which case it's about 73. So the place this is significant is really for skeletal muscle during heightened activity. So if you're running or riding your bike or working out and you feel your muscles, you'll feel them physically heat up. And that, combined with the acid they're producing, allows the skeletal muscle mitochondria and cells to demand more oxygen from the blood that they're receiving. So BPG, I mentioned, was a consequence of glycolysis, and that is true. And because this chapter does not focus at all on glycolysis, I'm going to omit a bunch of steps here. So this is kind of the Cliff's Notes version. Quick and dirty version of glycolysis does not acknowledge all the various enzymes that it requires. So it's just an intermediate on the way to producing the byproducts of glycolysis. But BPG happens to influence hemoglobin's affinity for oxygen as well. So as BPG concentrations increase, implying glycolysis is happening at a higher rate, more oxygen is released from hemoglobin. So why are we focusing on glycolysis specifically? Well, answer me this. Do red blood cells contain mitochondria? Yes or no? Nope, they do not. You guys are correct. Very good. Yeah, so remember, when red blood cells are developing, they have organelles for a while, and that's only to build hemoglobin. And then once they're done making their hemoglobin, they just barf out all their organelles and become this weird little frisbee with no organelles and a bunch of hemoglobin. So because of that, their only way to do metabolism is glycolysis, because glycolysis happens via enzymes in the cytoplasm. So what might cause red blood cells to do glycolysis faster is the question that we have to ask now. Any hormone or cell signal that increases metabolic rate in general is going to increase metabolic rate in red blood cells, thus causing an increase in bisphosphoglycerate. So thyroid hormone, most cells express thyroid hormone receptors, right? So that's going to cause an increase in metabolism in red blood cells, and that creates a situation where more BPG is produced, which means that more oxygen is dropped off of hemoglobin. And if you think about thyroid hormone and metabolism, and also growing, for example, thyroid hormone causes an increase in metabolism in your body globally, which means that your oxygen needs globally are going to be increased, which means that you're going to need more BPG to cause hemoglobin to release oxygen at higher rates. So it helps keep the demand for oxygen and the supply of oxygen off of hemoglobin matching. which is pretty amazing. So basically, its role is to improve oxygen delivery. Okay, so we have a few slides left, and I want to break the video up into smaller chunks, so I'm going to have another break right here.